All right, guys, I'm going to go ahead and get us started. My name is Kimberly Richardson, and I am a research advocate at the University of Illinois Cancer Center. And today we are here with you to talk about our project, Survivors Advising Scientists Educational Program. But our topic today is a collaboration that amplifies the importance of patient advocacy. I'm here today with MD PhD students Tova Bernstein, Casey Blaha, and Benjamin Gordon. Next slide. Morgan, you can go ahead and give those housekeeping notes if you like.
Hello, everybody. These housekeeping notes are just here to say that you will be muted. Your webcam will not be shared. If you're having some unstable internet connection, feel free to dial in using your phone. There's an invitation. On the invitation you got through your calendar invite, there's a phone number which you can call, and that'll dial you into the webinar. Any questions you have for the panelists, please send them in through the Q&A box on your attendee menu. And if you have any questions regarding technical issues or for any answers to the polls that we're going to be having, you can put those in the chat. And like I said, there will be polls, so be on the lookout. They will pop up on your screen once we get to that part of the webinar.
First off, I want to make sure I say thank you to the Society of Integrative Oncology, our communications committee, for inviting us here today. To have this conversation and interactive dialogue with you around our educational modules. I wanted to start here briefly with how this whole thing came to be for me as a patient advocate. In 2013, I was diagnosed with a rare form of ovarian cancer, granulosa cell tumor. Didn't know a lot about it, both in 2013, not a lot was on the internet, and unfortunately, my communications with my oncologist weren't all that great. So there wasn't a lot of shared decision-making, and there wasn't a lot of education for me as a patient. And so when I started getting involved in advocacy, the one thing I wanted to do is make sure no one else had my experience, right? That when they come to their oncologist and they've gotten this diagnosis, that they have a little time to breathe, but as well be able to go and find resources and start asking questions and getting answers to questions and developing better questions when they actually have to see their oncologist for upcoming visits. So I've began some work at the UIC Cancer Center. And in the middle there is what we consider to be community to bench. In this slide, in this photo, this is actually an epidemiologist who used to work at the cancer center. And I saw her in the hallway and said, can you help me with something? I as a patient, I am having the hardest time getting Sears registry data on ovarian cancer and really getting a real sense of what are our numbers in the state of Illinois. And for years as a patient, couldn't get that information. I think I talked to Kathy about that around 9:30 in the morning, and by 1:30, her student intern had given me all the data that I needed. And so imagining that that's what this means, more importantly, to find out that I was going to be a part of a presentation that she was doing around our vision of community to bench at the UIC Cancer Center. And so that last photo there is a little bittersweet for me, because the gentleman on the right is Dr. Robert Nguyen. He was our past executive director, and Dr. Kidieski is now our present Cancer Center director, and both had the vision for making sure that community means the patients that the UIC or UI Health actually serve. Next slide. And so what we did as a person who was starting to do more work at the Cancer Center, Dr. Kidieski came and spoke with a bunch of cancer survivors and invited us all to his cancer biology lab. And at the time, we were called the Patient Brigade. That was a term that Dr. Nguyen had given us, a little term of endearment. And you'll see us all there, Dr. Kidieski in the back. But most importantly, we were able to talk with all of the cancer trainees in his biology lab and ask really important questions and find out the type of research they were doing, even getting an opportunity to look at their cool new equipment. But what's the best scenario out of this picture is I am actually standing behind the cancer trainee who, upon my leaving, tapped me on the shoulder and said, how can we do more work together? This was like a super cool lab visit, and we want to figure out ways that we can do more together. Next slide. And so this is really how Survivors Advising Scientists educational program actually got started. I, as a patient advocate, was a part of the National Coalition of Cancer Survivorship, and they had come up with a program called Elevate. And we all had to apply to be an Elevate ambassador. And we all had to come up with an idea that we wanted to do, and NCCS would help us with a strategic plan. And so I had created an idea in my head about patient advocates collaborating with scientists. And this is literally how these training modules got started. Next slide. So in essence, this is what we do. It's a very bi-directional workflow. And what we mean by that is super important for everybody here on the call to really get and understand. How this starts is that the UIC cancer trainees, they develop the educational content of the module. The whole goal is for it to be easy to understand. But here's where it gets to be bi-directional. Then it's sent out in its draft form to patient advocates nationwide. And a lot of the patient advocates that have actually reviewed our modules are on the call with us today. Thank you so much for your support. They give them every kind of critique. I mean, literally from, we don't like how many times you say, like I'm doing. Everything from their diction to how the slides are presented, how the terminology is explained. But the super cool part about it is that the trainees then come back in written form to the patient advocates and say, I get what you're asking me for, I can change this, but here are some things that I possibly can't change. Can you help me figure out a way to communicate the things that I can't change in a better way, right? And so they create the content, it's reviewed by the patient advocates, advocates get more feedback, and then we share those modules out to the public. Next slide. So here's the real deal, guys. This is the reason why it's important to start collaborating with patient advocates. COVID-19 showed us nothing more. If it didn't show us anything, it showed us that it's the importance of explaining science in easy to understand language, and how influential that actually was on the general public. And in terms of our audience, there are like two different places where patient advocates really share our story. And it's either through legislative policy, and it's also through marketing in the media. It's going to be the same for your cancer researchers and your junior and senior researchers. At some point, it's going to make better sense for the trainee as well as the senior researcher to be able to convey their research in easy to understand language so that we can better communicate it and get you more funding. So it's an expedient transaction relationship, but I think what you're also going to learn is that the more you work with patient advocates, the more of our humanity comes out between both ourselves and the researcher. So next slide. So this is some of the things that I'm actually able to do. And I want to make sure that people understand that this is not me touting what I do, but that I'm actually leading by example. In cancer centers, I get an opportunity to be a part of the UIC's gynecological disease team, their working groups, tumor board. I get to give perspective on education and training of MD, PhD students. In terms of the private sector, I get an opportunity to be on patient design studios and be a patient expert counsel person in terms of trial design, apps, wearables, that whole continuum from the beginning to the end of a trial. And of course, with government, in terms of task force, I sit on the NCI's Moonshot program. I do DoD grant review, and I'm influential here at the state level in terms of policy change. Next slide. So this is what we really need to think about for both of us as advocates and researchers. We need to overcome these implicit biases. For researchers, I'm putting these questions out there for you. Please have a deliberate conversation in the chat about these issues. What skills do you believe a patient advocate needs to collaborate with you on a research project? And advocates, if you were asked to collaborate at the start of a research design, what skills do you think you need to have to participate? And then largely and lastly, both of us, what is a meaningful collaborative effort? It's not meaningful when you ask us in the 99th hour just to read an abstract that you're about to put in for a grant. We really want to be a part of the process. So is it in terms of a research design, patient engagement on a study, or even a journal article? Next slide. And so now I'm going to lend this over to Casey Blaha, and she's going to start to walk us through what exactly we mean about our bi-directional educational program. Casey?
I think Ben will introduce for us.
You know what?
first.
I'm so sorry. That's correct. Ben,
I
sorry about
usually
that,
go.
Ben.
We changed it.
Okay.
No
Thank
worries.
you, guys.
Hello, everyone. I'm Ben, and again, I just want to thank Morgan and everyone else at the Society of Integrative Oncology for having us. It's such a pleasure and honor to be able to speak to everyone and to start this conversation. So first, I'll give a little bit of background before I jump into some of our video modules and explain and show them off. So I am a fifth-year MD PhD student at the University of Illinois at Chicago. The MD PhD program, otherwise known as the Medical Scientist Training Program, is an 8-year program where you receive both your medical degree and a PhD. So our goal here is to become physician scientists. So doctors that see patients, but also perform experiments and run a lab. And Kimberly kind of already hinted at how we met, but we met in the fall of 2019. There was a picture of us standing next to each other. And it's kind of funny looking back at that. She's standing right behind me. Just to look at it, we barely knew each other then, but just to see, the partnership that would follow after that meeting. So as a MD PhD student, our ultimate goal is to serve patients. That's why we train in medicine. That's why we train in science. But when you're in the lab working on your PhD, you're kind of in, these fancy benches with a bunch of fancy equipment, but you don't really see patients at all. And, that's kind of this gap that is kind of unfortunate because we are working on cancer-related projects with the ultimate goal of helping patients. So it was really transformative to me to meet all these patient advocates and to start this conversation. And it was really impactful to me, the amount of curiosity, the true and deep concern everyone had, and motivation. And I thought this was an incredible opportunity to start a partnership. So I reached out to Kimberly, who had this great idea of SAS EP, which would eventually become. And as Kimberly mentioned earlier, it's this bi-directional partnership where we, as students, try to share our scientific knowledge, but we also learn a lot in the process about scientific communication. So it's learning and teaching on both ends in this partnership. So Kimberly already kind of showed the workflow of how it works. So now I'm kind of going to jump into the actual material. So we kind of organize the video content in terms of how they're kind of organized into groups of videos where they kind of follow a certain topic. So we have our first modules were on scientific designs. We taught what hypotheses were, experimental design, variables, biases. Then we also moved into basic science concepts, the science of cell biology, cancer biology, which we're going to show here. Actually, there's elements of both series. So we're kind of showing off some of the first ones, some from the second one. So you'll see a bit of both. We're also moving into a more clinical direction and gynecological cancers. We're also going to move into clinical trial design and all of that. So we're really trying to cover everything. There's kind of, no ceiling. There's so many different topics we want to cover. And the basis of this program is to get feedback from patient advocates and hear what they want to hear. So we have this email, you can see at the bottom, it's sasep@uic.edu. So we're always welcoming more feedback, more ideas, and we want to keep the dialogue going. Next slide, please. So here is one of the modules put together by Tova Bergston. This is on cancer hallmarks, part 1. Hallmarks is a term to kind of talk about what makes a cancer cell, and it's the distinct characteristics of what a cancer cell is. So we're going to press play. We're going to go over one of the videos. It's not too long, like 5 minutes or so, and then we'll move on to some poll questions after.
Hello, this is Tova Bergston, an MD PhD student at the University of Illinois at Chicago College of Medicine. This video is part of our Survivors Advising Scientists educational program, a bi-directional educational platform meant to connect our communities and increase scientific literacy and engagement. Our goal is to bring everyone to the table for scientific discussions related to cancer science and therapies. This is video number 5, Cancer Hallmarks, Part 1. There are many types of cancer that all have distinct properties and characteristics. However, roughly 10 characteristics have been identified in a majority of cancers, and these have been called cancer hallmarks. These hallmarks allow cancer cells to stay in the body and spread disease. Each hallmark is a complex topic, but we'll briefly cover the most well-known hallmarks in this video series. Typically, it is thought that these hallmarks can accumulate in a pattern, starting with changes that allow cancer cells to grow unchecked, followed by changes that allow cancer cells to be better at surviving when they should not. The first hallmark we'll discuss is somewhat unique and covers the ability of cancer cells to change their cellular makeup to allow the persistence and progression of disease. We suggest viewing video number 4, called When Things Go Wrong, to understand more about this complex topic, which we'll cover only briefly here. This hallmark is called genome instability and mutation. The genome is all of the DNA that makes up the contents of a cell and causes its functions. Cancer cells can develop instability of the genome, which allows their DNA to become changed or mutated. This ultimately gives them helpful properties that normal cells do not have. We can think of this as our cancer cell car having a mutated gas pedal, which is extra sticky, or brakes that don't work, which make the cancer cell car go faster and farther than the normal cell car. The next set of hallmarks we'll discuss focuses on cell growth and division, which are a critical part of normal, healthy cell life. However, cancer cells can have hallmarks which help them escape the normal mechanisms that control growth and division. These hallmarks allow the growth of cancer cells to become unchecked and go haywire. One of these hallmarks is the ability to sustain proliferative signaling. Most normal cells in the body are made to stop growing or proliferating when they have reached their full potential. Even when normal cells reach this point, cancer cells can keep proliferating. To demonstrate this point, let's say this car is a normal cell and the bottom car is a cancer cell. To stop the cars from moving, normally they would get a red light. However, the cancer cell gets a green light so it can keep growing. Another hallmark in this section is the evasion of growth suppressors. Normal cells can have their growth stopped in multiple ways. Cancer cells have developed ways to escape these methods and keep growing when they should not. We can think of this as our cancer cell car developing the ability to escape or turn off red lights so that it can continue to drive and grow. A similar hallmark is the ability to enable replicative immortality. Normal cells reach a point where they are no longer able to multiply or replicate, and they die. Cancer cells, though, have the ability to grow without ever reaching this point and are basically immortal. We can think of our cancer cell car in this way as only ever seeing green lights, never being told to stop driving or growing. The last hallmark in this section is deregulating cellular energetics. Normal cells use energy in very specific ways, but cancer cells can use energy in different ways that help them grow faster, multiply more, and even take nutrients from nearby normal cells. We can think of this as our normal cell car using just one type of fuel, while our cancer cell car can use more fuel that might be more efficient and gives it a growth advantage. Thank you for viewing video number 5. Please be sure to check out this video's part 2 up next. We'd also like to thank all those involved in the creation of these videos, including the University of Illinois Cancer Center and its Patient Brigade for providing feedback and shaping the direction of these modules. We invite you to reach out to us at sasep@uic.edu if you have any questions or feedback you'd like to share. Thanks again.
Okay, so that was an example of one of our video modules that Tova put together. Now we'll just, to make this fun and engaging, we're gonna just follow up with some poll questions. So I believe you answered the poll via the poll number 1 in the attendee menu. Oh, I think it just popped up. So you're not gonna be graded at all, and I don't think anyone will see your specific answers. So, but please try to answer these to the best of your abilities. And in a minute or so. And then after that, we'll review all the questions together. Okay, so looks like we were perfect. So everyone answered the correct questions, but let's move on to the next slide so we can go over them one by one. Okay, so question number 1, true or false? Cancer cells can escape normal growth mechanisms and grow unchecked. That is true. As Tova explained in her video about cancer hallmarks, mutations for cancer cells typically allow them to grow more, not less. So very good, we got that one right. And according to Tova's examples with the cars, we thought we'd put a car example. So an uncharged electric car or a motorcycle without gas, a taxi cab with flat tires, both wouldn't be able to go very far. But a sports car and acceleration that does not have any brakes speaks to what a cancer cell is the most. So good job, everyone. It looks like everyone was perfect. And I believe I'm handing the mic off to Casey at this point. So thanks, everyone.
Hello. I got a brief hi earlier, but I'm Casey. I'm also in UIC's MD PhD program. I am in my last year of the program, actually, which is kind of fun. I defended my PhD last year in cancer metabolism, specifically looking at breast cancer metastasis. And actually, around the same time when I was defending my PhD, Ben had reached out to some other of this MSTP students talking about this project. And I was in kind of this unique position because for your thesis defense, which I actually ended up doing via Zoom, you prepare a presentation to family and friends and not just your colleagues in the research field. So I was in this trying to make a presentation on my scientific research that also can be relatable to my family and friends. And throughout this project being involved in this project has really given me the experience in trying to, like Ben was saying, really learn how to better communicate as a young growing physician scientist. And in the last years of the MD PhD program, you go back to medical school, and I'm in the clinic, in the hospital. And I think really something that I found is becoming a part of my practice is really focusing on patient education and as well as patient advocacy. So I think being involved in this program has really kind of put me on a new, not just cancer research pathway, but also continuously being involved with patient advocates and patient education. So that's my little spiel. Sorry, I lost my voice a little bit, but I'm going to continue with the modules that we've created. And we will talk about Cancer Hallmarks Part 2. Again, they'll have a brief 3 questions that just kind of quick quizzing on the basic knowledge that you'll gain. No pressure. It's just to kind of see what you can gain from kind of what we hope you can gain from watching these quick, short modules. So thanks for participating.
Hello, this is Tova Bergston, an MD PhD student at the University of Illinois at Chicago College of Medicine. This video is part of our Survivors Advising Scientists educational program, a bi-directional educational platform meant to connect our communities and increase scientific literacy and engagement. Our goal is to bring everyone to the table for scientific discussions related to cancer science and therapies. This is video number 6, Cancer Hallmarks Part 2. Please make sure you have watched Cancer Hallmarks Part 1 before continuing with this video. There are many types of cancer that all have distinct properties and characteristics. However, roughly 10 characteristics have been identified in a majority of cancers, and these have been called cancer hallmarks. These hallmarks allow cancer cells to stay in the body and spread disease. Each hallmark is a complex topic, but we will briefly cover the most well-known in this video series. Picking up where we left off in Part 1, we will continue covering the remaining hallmarks in this video. This set of cancer hallmarks are properties that help cancer cells move to other parts of the body to spread disease. The first of these hallmarks is the ability to induce angiogenesis. Angiogenesis is the growth of blood vessels. Normally, blood vessels are found all throughout the body. Their main purpose is to help deliver necessary nutrients, like oxygen, to tissues to help them grow and survive. They also allow cells to travel from one place to another, like when helpful immune cells travel through the blood vessels to reach the site of a cut or infection. Cancer cells can cause new blood vessels to grow. This helps them by giving the cancer cells access to more nutrients from the blood and providing them with a way to get to new parts of the body. We can think of this as our cancer cell car finding a new gas station for fuel and new roads to travel on. A similar hallmark is the activation of invasion. Invasion is the process of moving from one site or tissue into another. Most normal cells are not capable of invasion and stay in organized layers in the tissues where they belong. But cancer cells can cause themselves to invade through their original site of growth into a new site, helping it to spread throughout the body. We can think of this as our cancer car leaving its home city and traveling to a new one. The next set of cancer hallmarks are those which help cancer cells escape normal mechanisms that the body would use to stop or kill them. One of these hallmarks is the ability to resist cell death. The body can recognize when normal cells are unhealthy or not working properly. It can then cause these cells to die so that they can't cause problems. But cancer cells can escape or resist these mechanisms and stay alive even when they should not. This helps the cancer cells remain in the body to grow and cause the disease to persist. We can think of this as our cancer cell car being able to avoid getting sent to the junkyard and being able to drive free instead. A similar hallmark is the ability to avoid immune destruction. The immune system is a complex network that defends the body against illnesses like infections. Normally, the immune system can recognize dangerous cells and dispose of them. Cancer cells, however, can hide or escape from the immune system, trying to arrest them. This also allows cancer cells to stay in the body and the disease to persist. We can think of this as our cancer cell car being able to resist the police trying to catch it while it escapes to freedom. The final hallmark relates to the ability of cancer cells to be supported by inflammation. This hallmark is called tumor-promoting inflammation. Inflammation is a normal part of the body's response to fighting disease and infection. However, cancer cells can sometimes use elements involved in inflammation to help them grow and survive. For example, cancer cells can corrupt immune cells into helping them survive instead of arresting them. We can think of this as a normal immune cell, labeled here as police, causing traffic lights to turn green so they can pass through quickly, and our cancer cell car as taking advantage of these green lights to pass through as well, instead of getting stopped by the police. The hallmarks of cancer are quite complex, but they help cancer researchers learn how best to fight disease. In fact, many scientists are developing therapeutics or medicines that address one or many of these cancerous traits. This is just one way in which research aims to help cancer patients, and since the field is still working to understand cancer, we may even find more hallmarks in the future. Now we'd like to go through some questions that you can think of when you come across scientific literature on cancer properties. In scientific papers, you can ask, which hallmarks are the researchers investigating? How do different cancer medicines work to stop some of these cancerous properties? Thank you for viewing video number 6. We'd also like to thank all those involved in the creation of these videos, including the University of Illinois Cancer Center and its patient brigade for providing feedback and shaping the direction of these modules. We invite you to reach out to us at sasep@uic.edu if you have any questions or feedback you'd like to share. Thanks again.
Awesome. So we finished the 2 modules, part 1 and part 2. The next set of questions will be 3 questions that will just be focused on part 2 in a similar fashion as to what we just discussed. And again, they're popping up. And just a brief reminder while you're looking at it, these videos right now in this webinar, you have no choice but to watch them all the way through, but online you can pause them at certain times and look at slides on your own if you think if sometimes it's going too fast. So just take your time and answer these questions, and then we'll go through them. There we go. It automatically pops up. And everybody did great again. Yay. They weren't supposed to be tricky. And then the next slide will just be briefly touching on them. So the first question was, what was angiogenesis? Everyone correctly said that it was the growth of blood vessels, which provide nutrients and new routes for cancer cells to travel. The analogy we used were the new roads for the cars. Then we also had growth of lymphatic vessels, which actually are vessels that immune cells can travel on to help clear toxins or waste. This could in itself be a separate module discussing about the lymphatic system. Then the next question would be, which of the following is not a hallmark of cancer? Staying still, correct? The normal cells will stay in one place most of the time versus cancer cells will invade or metastasize and spread throughout the body. And then true or false, when normal cells are unhealthy, the immune system can make them undergo apoptosis or cell death. True, and in fact, normal cells, they will undergo cell death, whereas cancer cells have the ability to evade cell death and invade the immune system, invading the police or invading, and in fact, even using inflammation sometimes to its advantage. So thank you for kind of going through these two example modules that we were creating. The next part will be TOVA in another example module that we really like as a team.
Hi, everybody. I just want to second the thanks for having us here today, SIO, and thank you all for attending. My colleagues have pretty much hit on the head why it's really special to be a part of this program. It's not often that we're given the opportunities as researchers to work this closely with patients, but it has been incredibly impactful for all of us. And I think one of the nice parts for Ben and I, we started this at the beginning of our PhD. I think it's really shown me how important it is to seek out these engagement and communication with patients, because that will really help us become better physician scientists, because we're trying to serve patients, and how can we do that if we don't know how to speak to you? So it's been a pleasure, and one of the things we do a lot to communicate our science as researchers is present scientific posters. So you'll see them a lot if you go to conferences and things, but this little video will show you how to read one if you ever encounter one. So we can go ahead and start.
Hello, this is Tova Bergston, an MD PhD student at the University of Illinois at Chicago College of Medicine. This video is a part of our Survivors Advising Scientists educational program, a bi-directional educational platform meant to connect our communities and increase scientific literacy and engagement. Our goal is to bring everyone to the table for scientific discussions related to cancer science and therapies. This is video number 9, how to read a scientific poster. Before we start breaking down a scientific poster, we need to know what one is. While researchers typically aim to publish their work in written articles in scientific journals, a scientific poster is a visual way of representing research. Posters are typically presented by researchers at conferences during poster sessions. These are large gatherings where attendees of the conference walk around at will and can stop to listen at posters that interest them. At the posters, researchers will give more casual presentations of their research than one would find in a scheduled talk. Asking questions during poster sessions is highly encouraged. They are a great opportunity to engage with researchers. Now, the first thing we'll need to know about reading scientific posters is the direction. Posters are read from top to bottom and left to right. Typically, columns are used to direct your reading. For example, on this poster here, we would start with the green star and follow the black line down, across, and down until we finish at the blue star. There are many components to a scientific poster, and we'll go through each one individually, starting with the title, authors, and institutions. The title is meant to draw you in. It should give a succinct description of the project and basically tell you what is being studied or what the researchers learned. Next, we see the authors. The first name listed is typically the person presenting the poster, and the last name listed belongs to the principal investigator. The principal investigator is typically the head of the lab which is doing the research. Next, we see institutions, which are typically listed for informational purposes, in case you'd like to reach out to these researchers at a future date. Next, we'll look at the introduction or background section. This section is very important. It should be a brief background on the disease of interest. It helps lend significance to the work being done. It should cover something that is unknown in the field and should mention how this work is attempting to solve that unknown problem. The last thing you should find in this section is the hypothesis, or the scientific question being tested. Hypotheses are one of the most important parts of research, and you can learn more about them in modules 1 and 2 of this program. Next, we look at the methods section. Not all posters will have a method section. Sometimes this section is filled with the first set of data instead. But when a method section is present, it explains how certain parts of the work were accomplished. It is usually included when the development of a method is critical to understanding the work, or developing the method was a new discovery that is beneficial to the field. The largest portion of a poster will be the results section, which is often filled with data and figures. This section holds the answers found by researchers during their studies. A results section is always included in abstracts and journal publications, but on posters, you may not see a specific section titled Results. Instead, you might find a group of labeled figures. There are different types of data and figures, the most common of which is graphs. Usually, bar graphs will have a dependent variable, which is displayed on the y-axis. The dependent variable is representative of the outcome that researchers were curious about. The independent variable will live on the x-axis, and this is what researchers change in order to see an effect. You can learn more about experimental variables in Module 3 of this program. Well-designed experiments have controls. They are usually the leftmost bar in a graph. I'd like to go through an example of a control. If you are testing a new drug and want to find the best dose, the liquid or solution in which you dissolve the drug would be a good control. That way, you can make sure you're comparing the effect of your drug to a baseline of the non-removable effect of your solution. Another important thing to look for on posters is significance. Significance is determined by the p-value, which is obtained through statistical analysis. This is very complicated, and if you'd like to learn a little more, you can listen to Module 6 of this program. But for the sake of posters, scientists usually consider significant results as those with a p-value less than 0.05. If results are significant, that means it is unlikely they have occurred by chance, and they are more likely to be due to the change in the independent variable. Significance is represented by asterisks on graphs. These are seen as little stars above the bars. The next thing we're going to discuss is usually found in the bottom right-hand corner of a poster: future directions and conclusions. The future directions section is typically filled with work that still needs to be done, either to fully confirm or deny the hypothesis, or to answer new questions raised by the current research. Conclusions are typically a brief summary of the work that was done by the researchers and what they discovered. It usually includes whether or not the hypothesis was confirmed, and this likely informs how this work might be used in the future to continue to address the unsolved problem mentioned in the background. This is a great place to look if you want a quick version of the research that happened on this poster. Other things you might see on a poster, just so you're aware. References. These are publications that either formed background reading for this research or include the work shown on the poster. They're often useful for wanting to learn more about a topic. If you'd like to look at a reference at a future date, you can copy down the first author and the title. Then you can search for it on a database called PubMed or Google Scholar. You might also see something that says this work has been funded by. This is a reference to funding agencies where grants have come from that fund the research in this laboratory. Now, we'd like to go through some questions that you can think of when you're trying to read a poster. What were the researchers trying to accomplish? What unsolved problem were they trying to address? What is their hypothesis? For example, do the researchers think a new drug is more effective at treating a disease model than the current gold standard treatment? Is their data significant? Can you see statistics or p-values or asterisks? Are the results meaningful? What conclusions can you draw from this data? How might this data answer the unsolved problem? Did the researchers address their hypothesis? Was it accepted? Do there appear to be any apparent biases or mistakes or errors that could affect the results of the study? This is the end of Module 9. Now, the presenters would like to thank Dr. Kidievsky and the administrative staff of the University of Illinois Cancer Center for their support in this effort. Additionally, we'd like to give a special thank you to all the cancer patient advocates who provided feedback during the development of these modules. We'd also like to thank you for reviewing this module, and we invite you to send any feedback on this module or suggestions for future content to sasep@uic.edu. Thanks again.
Okay, thanks for listening to that. If we could go to the next slide. Awesome. Okay, so we only have about 9 minutes left, so I'll try and go through these really quickly. But if this were a scientific poster you encountered at a conference, if you want to type in the chat where you might start reading, but for the sake of time, I'll just remind you quickly that we typically start at the top left of a poster and work our way down toward the bottom right. Which graph has significant data? That's a big thing that we should pay attention to when watching scientific posters. And these are typically ones with little stars on top of them or asterisks. Some people will put multiple if the result is extremely significant. But so it's a little small here, so I won't make you name those either, but there are a couple. And then where can I see what the data means? So often this is in the conclusion section, which is at the bottom right. That's classically where people will take the results, the science, and make it into a sentence that actually means something to us as researchers. So that's all of that. I think we can go to the next slide, which we already covered, so we can go even one more. And then we just have a couple closing questions about SIO to tie in our webinar with ones that you've had in the past. So if you can think back to April, if you were present for that, one of the ways you can try and combine the things that we are talking about today, cancer hallmarks, with that talk, which was about dietary changes, such as very low intake of fat and carbs, how could that affect cellular responses, such as the cancer hallmark resistant cell death? And if you have any thoughts on that, you're welcome to paste it into the chat. And for the sake of time, we'd also love to hear from you what other questions you would like to ask your oncologists or support groups at the next webinar after these videos, based on the knowledge you all already have about integrative oncology. Feel free to share that with us as well.
If we don't have any general questions, I know that Eugene put a question in the chat. So Eugene, if you're still on the line, if you wanted to process that question with the panelists, that'd be great.
I can see it if you'd like.
Yeah, I could read it out loud if
Yeah,
that
sure
would be
thing.
helpful. Okay, so one of the influences patient advocacy has done is to increase the importance of quality of life as an endpoint on clinical trial validating or not any new drug. Would you agree that in the future, when determining pricing for drugs, we should factor in not only the impact of drug on survival, but also quality of life before determining its value to patients? And yes, the short answer, right now I'm performing basic science research experiments. I'm not well trained yet in the clinical world. After I finish my PhD, that's when I go back to the clinic, and I'm really looking forward to learn more about clinical trials. But I think this is one of the fundamental aspects is when doctors and scientists are designing these clinical trials and testing these drugs. I think it's really important. The experiment's only as good as what you have in mind when you're starting it. So if the scientist is only thinking about length of survival or overall survival or disease-free progression, and they're not even thinking about quality of life, then maybe the patient's concerns are not even being addressed at all. So I think this is where patient and advocates really deserve and need a spot at the table when these experiments, before they even start, when they're being created. So when the experiment's created or the clinical trial designed, patient concerns are directly ingrained right in there from the start.
I'd like to add a few things too, because I mean, even from the other side of it, I mean, the types of patient advocacy around the policy side of how drugs are being priced. I mean, to the extent that there are some pharmaceuticals that buy up smaller biopharmaceutical companies and just get a drug without even having to do any of the heavy lifting, but they could double the price on what that drug actually would cost for the patient. And so whether or not there have been any improvements to that drug after the purchase and takeover of these smaller pharmaceutical companies, nobody's looking at whether or not there's any improvements to that drug. At the same time, we're the ones that have to end up paying for it. And so it's a lot of things to look at in terms of patient advocacy. We have to look at the toxicity of these drugs without us having any input in the design of the trial. But then on the other side is it's the dollar and cents scenario of why the pricing is the way it is. If we are not watching and being the sort of watchdogs around advocacy for not only the research advocacy, but also the policy advocacy and being able to speak to those kinds of things in regards to price. So we are paying both with our bodies as well as in our pocketbooks. So thanks for the question, Dr. Ahn. Are there any other questions about the modules? Because we really do want to get that sense from you that, they're not too oversimplified, but that they do speak really well to the patient and the caregiver who's just recently either been diagnosed or about to stop treatment. Because the whole idea behind the modules are to empower patients and to give them a sense of what advocacy is actually like when you start talking about whether or not you'd like to be a research advocate. Because there are lots of lanes in advocacy. And I think most of the people on the line who are patient advocates know that. And it's really simple to be in awareness advocacy. You just get to share your story. Sometimes with policy advocacy, you do get the chance to share that story and go to the Hill or go to your state representatives and talk about the importance of cancer research. But until all of us, patients, researchers, caregivers, really understand the language behind the research, we can get stuck. I remember very distinctly being in a conversation in DC, and one of the staffers asked one of us in the ovarian cancer group, well, tell me more about how PARP inhibitors are working for ovarian cancer. And the person he addressed that question to, she wasn't necessarily as involved in research advocacy, to the extent that I was able to swoop in and say something, right? But we need to be so much more equipped as patient advocates when we're talking very importantly to appropriations committees about the importance of funding research. And here with integrative oncology, I think it's so critical that as patients come through their diagnosis and through treatment, that they understand more about our modalities within integrative oncology. And if we should be able to put some modules together with cancer researchers and cancer trainees, along with the reviewers of patients from our neck of the woods, so to speak. Thank you so much for that, Mel. Are there any other questions? Well, we are right at the top of the hour. Next slide.
I was like, we have a slide, but they're in real, I couldn't remember if we put it in here or not, but kind of just to follow up what Kimberly was saying. And I think also in the Q&A, just please email us with any suggestions of new modules that you would like to be created. The email we just posted to the chat, either just creating in general, but also interested in being involved in either as a reviewer of the module, or we're also looking for content experts. So people that can help us in the initial draft form, throughout any of the process, either examples of, we threw some out here, there's some in the Q&A, so feel free to reach out to us. We want to keep expanding these modules and hope that we can keep working together to move forward. So thanks for coming to the webinar.
I don't have anything else to offer other than to thank you all for the opportunity to come and chat with you guys about the modules. I'm super proud of my colleagues within the MD PhD students, because they are going to be the future. They're going to be those senior researchers one day, and they already get the concept of collaborating with patient advocates. And I'm just so incredibly grateful for the opportunity to work alongside them. So thank you guys. And thank you all for giving us this hour and the opportunity to share our modules with you. Morgan, we're ready.
Thank you, everybody. Everybody have a great day.